Thank you, and thanks for inviting me here. So, or here, I didn't go anywhere, I guess, but okay, so today I'm talking a little bit about universal properties of strongly coupled networks. This is the term that's used in the literature for it, but really what I mean is not so weakly coupled networks, and I'll clarify that in a little bit. They're not actually all that strongly connected. So the idea is that cortical neurons receive hundreds or maybe thousands of synaptic inputs from other neurons, either in other layers or other cortical areas or subcortical areas or from within their own layer. But postsynaptic potentials, each postsynaptic potential pushes the membrane potential, around 1/30th or 1/50th or so of the distance from reset to threshold. So in this sense, the networks are not all that weakly coupled, right? They receive far more inputs than are needed in terms of the scale of the distance from rest to threshold. So to formalize that mathematically, just consider a large network. For now, let's not even specify how the membrane dynamics or firing rate dynamics evolve. Let's just consider a large number of excitatory and inhibitory neurons that are recurrently connected and receiving input from some external layer. We use INs to denote the size of the networks. And then the synaptic input to one neuron, this would be neuron J in population A in this equation, is written as a sum of synaptic weights across other neurons. And then for a spiking network, you might have a postsynaptic current or postsynaptic conductance waveform summed over spike times. For a rate model, you would just sum the rates, right? So the idea is there's this inner sum, this sum over n. So in general, neurons can receive a lot of inputs because n are large, the networks themselves are large. More specifically, following from that equation, one would come to expect that the synaptic input to a single neuron is order j times k, where j is a typical synaptic weight, k is a typical n degree, or if we want to consider randomly connected networks, it'd be j times the probability of connection times the number of neurons. Of course, if there's multiple populations, there can be multiple probabilities of connections or n degrees from different populations, but this is the general idea. But we don't want I to explode, right? We want to consider large N, large networks, and we don't want the synaptic input to any individual neuron to become very large. So classical methods for avoiding large synaptic inputs are to consider sparse coupling. This would be taking connection probabilities that are 1 over n, or n degrees that are order 1 as n gets large. Or weak coupling, this would be scaling synaptic weights by 1 over n and keeping connection probabilities order 1. But we argued from the beginning that maybe cortical circuits aren't all that weakly or sparsely connected. That was my first slide, right? So the question is, can we maintain sort of reasonable magnitudes of synaptic inputs in large networks without assuming sparse or weak coupling? So why don't we just let this product of J and K be large and see where it takes us? So now let's just assume that neurons receive a lot of inputs that are not all that weak. So let's clarify here, though, we're not assuming that individual synaptic weights are large. We're not even assuming that they have to be order 1 as n grows, or you can assume here that n is growing or k is growing. We're going to grow these proportionally in the models we considered here. We're only assuming that they're larger than 1 over k, or larger than 1 over n, however you want to look at it. In other words, the network is not so weakly coupled, right? So we don't scale synaptic weights like 1 over k or 1 over n. And this is sort of borne out to some extent by some experiments in culture by Alex Reyes, who found that if you grow cultures of cortical neurons and increase the density of neurons, effectively increasing the number of neurons in the culture, the number of connections increases proportionally to the density. So that means the probability of connection is approximately order 1, but the strength of connections, so the postsynaptic potential amplitudes that they see, actually decay. So they get smaller as the network becomes denser and more neurons receive more inputs. But they don't become small like 1 over k. So the best fit power law of this is 1 over k to the power of 0.6. So I'll be more specific about that in a little bit. So balanced networks take a scaling where J does not scale like 1 over k, where jk is large. And balanced networks, if you're unfamiliar with them, synaptic weights scale like 1 over the square root of the n degree, 1 over square root k. which means that this JK bar parameter I've defined, which kind of quantifies how strongly the network is coupled in a sort of mean field sense, is order square root k. Or if we take n to infinity instead of k to infinity and fix connection probabilities, then the n degree is order n, k is order n, and the product of j and k, which we're sort of using to quantify the coupling strength, is order square root n, and we can take n to infinity. We're going to kind of consider these two limits to be equivalent because we're taking connection probabilities to be order 1. So as I mentioned before, I accidentally swapped the order of these two slides at the last minute without realizing I would scramble some things. But Alex Reyes showed that this is approximately true in cortical cultures. He found that synaptic weights scale like approximately 1 over k to the 0.59, whereas 1 over square root k would be 0.5. So the best fit decay of synaptic weights is very close to what you see in balanced networks, a little bit faster, but not 1 over k, right? The slope would be -1 if it were 1 over k, which would be a weakly coupled network. Finally, balanced network models, when you do scale synaptic weights like this, reproduce a lot of features of real cortical circuits. So you get asynchronous irregular spiking, you get spike train correlations that have a mean that's very close to 0 and are distributed in a sort of symmetric way around 0. This was first shown by Alfonso Renart and Jaime de la Rocha and their colleagues in 2010. You get EI tracking in the sense that excitatory and inhibitory currents, inputs to a neuron, sort of track each other. They're highly correlated. And E and I are anti-correlated if you take their sign into account, the polarity, which is what we see in a lot of recordings. We also showed that in these balanced network models, you get distance dependence of correlations. So the decay of these weak correlations with respect to the distance at which neurons are from each other is reproduced in the models like you see in experiments. Okay. So just to qualify this though, in reality, this JK bar might not be all that huge. Ahmadian and Miller recently argued in arXiv that it's not all that large. Of course, it probably depends on species. It might be different in mouse and primate, might depend on cortical layer, cortical area. Depending on how it's measured, this could depend on behavioral state or sensory state, something like that. But I just want to argue that the large JK limits, these not so weakly coupled networks or strongly coupled networks, if you want to call them that, provide a decent first approximation to cortical circuit responses. And today I'm going to argue that they provide a mathematically rich approximation, which is something that this ICMNS community presumably likes, and that it's mostly model independent. That is, we can look at the network architecture and understand about approximately what the network is doing without having to specify exactly how we're modeling individual neurons. So that's the argument I'm going to make today. So the first part of my talk, I'm sort of on a soapbox. Why should we be modeling circuits like this? And the last part of my talk, I'll talk about some results. Okay, so let's transition to the mean field. Let's say we have one of these not so weakly coupled networks. So the recurrent connectivity, the recurrent input to a neuron, so I here is the average input to each population in the network, is going to be order JK bar, like I said, and JK bar is going to be large. Similarly with the feed forward input, which I'm denoting with a subscript X here, this is the external input to the network. We can pull that JK bar out to kind of keep track of the fact that it's that it becomes large and everything else is order 1, all the other parameters. And it's long been known that in this limit, the strong coupling limit, that rates are approximately a linear function of external input. So they can be written as negative W inverse X here. The idea is that if JK becomes large and you want the inputs to not become large, you need a cancellation inside that square bracket right there in the first equation. I think I can actually point to this. You should be able to see my pointer here. In fact, you can prove this if you want. It's actually not that difficult to prove at all. If you want I to be order 1, then you need the limit as JK bar goes to infinity of R to be this function. So that's a very nice, simple framework. It applies to rate network models. It applies to spiking network models. It really all depends on just the linear algebra of this equation. It's not even a very complicated concept to prove. Also, it generalizes to any number of populations, so we don't have to consider just one E and just one I population, and to spatially extended networks. Here are some examples from So papers that I've worked on with my students and an old paper with my postdoc advisor where we tested this approximation in various networks. So here on the left is a network of exponential integrate and fire neurons with 4 populations, 2 excitatory, 2 inhibitory. In the middle is a network of adaptive EIF neurons with 2 populations, 1 excitatory, 1 inhibitory. And all of these plots, the solid curves are from spiking network simulations, and the dashed curves are from that equation I showed on the previous slide, or the equation that's right here in the center of this slide. And then the spatially extended example, with LAFs, we pushed the number of neurons up to 2 million. So we simulated 2 million LAF neurons and found that this, in the spatially extended case, even this approximation for R is pretty accurate, and the large N limit. So in all of these cases, we're looking at how accurate it becomes as N grows large. Okay, so there's some problems with this though. So this equation has been known in various forms for a while, but there are some problems with it. The first is that in this equation, X here is the external input to the network. So it's like a stimulus. W is the mean field connectivity matrix. So it's the matrix of average connection strengths, you can think of it, between neurons in different populations, right? And R is the vector of population average firing rates. So this tells us that the population average firing rates are a linear function of the stimulus, at least under this approximation, but we know the cortex can perform nonlinear computations, obviously, it needs to. Another problem is that under this equation, parameters need to be chosen to produce non-negative rates. Now that's relatively easy when you have one E and one I population. When you just have 2 populations, then you need to satisfy 2 inequalities that are relatively well known. They were in the original Van Riesbeek and Sopolinsky paper, and it's easy to choose parameters such that that's true. However, every additional population you add onto your network adds another inequality to satisfy, and the proportion of parameter space that satisfies all of those inequalities is then going to decrease exponentially with the number of populations. So it becomes more and more difficult to satisfy this inequality as you include more and more populations in your network. And my student, Vicky Zhu, was able to prove actually even more sort of damning is that for any connectivity matrix W satisfying Dale's law, there's a positive stimulus, a positive X, for which this expression, negative W inverse X, has negative entries. Remember, negative entries are things that break balance. That means any connectivity matrix that satisfies Dale's law admits a stimulus that breaks balance. So while simple and somewhat model independent, this equation has these weaknesses that means maybe it doesn't account for everything we sort of need it to. Okay, so to demonstrate these notions, here we simulated a network of 30,000 EIF neurons, 2 excitatory populations, 1 inhibitory population. They're receiving external input from 2 external populations. There's connectivity between all the recurrent populations and from the external to all the recurrent. And for the first 500 milliseconds of the simulation, you can see the raster plots of a subsample of the neurons on the left, voltage trace of 1 neuron. In this case, we put the network, we chose an external stimulus such that the balance state exists. And you can see everything sort of works out nicely. You get a balance between excitation and inhibition and the inputs to all 3 of these populations. But then we transitioned to a stimulus for which this balance state does not exist. So this equation that I wrote before, R equals negative W inverse X, predicts that some population, one population, E1 in particular, would have a negative firing rate. So it can't possibly be valid. And what we see in this case is maybe not so surprising when you think about it. That population turns off. It just doesn't spike at all. You can see that in the raster plot on the left. It receives excess inhibition, so it's breaking balance here. But the other 2 populations then form a balanced subnetwork. You can basically forget about population E1, and now we just have sort of a standard balanced network with populations E2 and I. So this is sort of an illustrative example of what I refer to as the semi-balanced state. So the semi-balanced state occurs when balance is broken. So when you have this balance equation predicts 1 negative rate, which gives rise to excess inhibition to some populations, and the remaining populations form a balanced subnetwork. Okay, and it's very simple when there's just a few populations, but it's actually more difficult to work out what exactly is going to have when there are more populations. So my student, Vicky Zhu, again, was able to derive an equation for firing rates that generalizes this W inverse X equation that I was talking about before for balanced networks. She was able to generalize this to the semi-balanced state with a couple more assumptions. So here we have to assume an Fi curve, but we only place relatively weak assumptions on the Fi curve. And we find that in the semi-balanced state, we get firing rates are satisfied by this equation here in the gray box, where the brackets plus is a rectified linear threshold linear function. So you can kind of see if you think about it for a minute, if R is positive, if WRX plus R is positive, then this is going to reduce to WR plus X equals 0, which gives back the same balance equation we had before. So this generalizes that. So when all the firing rates are positive, this gives the same state. But when some firing rates are 0, it gives a different state. This could admit multiple solutions as well, unlike the standard balance equation. Well, here's a more direct comparison. And the assumptions, the main difference in the assumptions between the two theorems is that for balance, on the bottom here, we assume that the input to all neurons remain bounded. For semi-balance, we assume that none of the firing rates approach the maximum of the Fi curve. So that could be infinite if you have an Fi curve that goes off to infinity, or it could be finite if you have a saturating Fi curve. So if you assume that you have a strongly coupled network, and you assume that none of the rates blow up and saturate, then the rates have to satisfy this equation and the strong coupling limit. Whereas for balance, we assume that the inputs remain finite. The difference is in the semi-balanced state, some of the inputs can diverge to negative infinity, which just turns off those populations. One thing that's nice about this equation is it gives you a nonlinear mapping from stimulus to rates. So now our networks can implement nonlinear functions, which was a weakness of the balanced state that they couldn't. Another interesting thing about this equation is that it maps directly to fixed point rates in RNNs with rectified linear activation functions, which are equivalent to these threshold linear networks that Karina Courteau and her colleagues study. So you can just adjust the connectivity matrix by adding one term, and you get back exactly what Karina Courteau studies. And she's shown that these networks can exhibit some very rich and interesting properties. So there's a lot to study here. And now we've connected that to any sort of strongly coupled network. It doesn't have to have a threshold linear activation function, and it can be a spiking network, whatever you want. And it provides a decent approximation for networks with large n. So again, we're always looking in this sort of large n limit, and biologically realistic coupling strength. So we simulated networks of adaptive EIF neurons, Izakevich model neurons, adaptive EIF with conductance-based synapses, for which we have to make some adjustments to the equation, but it still works out. And these based rate networks and a stabilized, super linear stabilized state studied by Ken Miller and colleagues. Okay, so it gives a decent approximation, and it's an interesting equation. So far though, I've only talked about balance and now semi-balance at the level of population averages, right? This is to say that so far I've talked about if you average the input to neurons in a population, the excitation, the average excitation to the population is going to approximately cancel with the average inhibition. But now let's transition to talking about what's called detailed balance, which is to say that every single neuron in the network receives a approximately balanced input. Okay, so to demonstrate this idea of detailed balance, I ran a simulation of a network with just one E and one I population now. And for the first, I think it was 40 seconds of this simulation, the left plot of this time series here, this was just a randomly initialized network, nothing special about it. And you can see here that excitation and inhibition, so these are each red line here, each red curve is excitation to one neuron in the network. I think these are strictly excitatory neurons, but it doesn't really matter. Each blue curve is inhibition, and the black curves are the total input. And you can see that the network is not really balanced, right? The spread of the total inputs is similar to the spread of the excitation and the spread of the inhibition. So it was shown by Tim Vogels and colleagues in 2011 that inhibitory synaptic plasticity, homeostatic inhibitory synaptic plasticity, can achieve detailed balance. So after 40 seconds of the simulation, we turned on inhibitory synaptic plasticity, and indeed, we see that the network achieves detailed balance. So all the neurons receive balanced inputs, right? And you also see that the rates sort of collapse to this target rate that's defined by this STDP rule. So this is a classic picture, well known by now. You can also see it quantified here. This is a histogram in log space, so with a logarithmic vertical axis of the total current. That's the black time series in the right plot. And before synaptic plasticity is turned on, you see this very broad distribution of total inputs. After synaptic plasticity is turned on, you see this very narrow distribution of total inputs, meaning that excitation and inhibition are precisely canceling on a neuron by neuron level. Okay, but this is for one input. This is for all the neurons receiving the same synaptic input, external input, sorry, one stimulus. So the whole network's just receiving a time constant stimulus. If we now start changing the stimulus, so what we did here, you can see in this network schematic to the left, what we did is we basically, every second, I think, or 2 seconds maybe, we randomly sampled a stimulus from, so the stimulus space now is n-dimensional, the dimension of the number of neurons in the network. Here there's 5,000 neurons in this network. We sampled a new point in this 5,000 dimensional space, but we restricted ourselves to a 2-dimensional plane. So we're basically sampling points in a square in this 5,000 dimensional space. And when we did that, what we see is we no longer exactly get detailed balance. So here, just looking at the time series first, we see these deviations in the total input, but only more or less only in the negative direction, right? So you see these negative deviations of the total input. And if you look at the distribution, that's very clearly borne out here in this heavy negative tail in this purple histogram here. So what that tells us is that this network is, and we also see firing rates that hit 0 every now and then. And this tells us that this network is in a detailed semi-balanced state, in the sense that you never receive excess excitation, but some neurons receive excess inhibition in response to some stimuli, and those neurons are silenced by those stimuli. And the stimuli change, and it's another subset of neurons that are silenced and receive excess inhibition. This is the defining feature of the semi-balanced state. So that is to say, networks with homeostatic inhibitory synaptic plasticity, if they receive input from, if they receive one stimulus, and this is actually the scenario in which they're very often studied, then you get detailed balance. And that's been known. But actually, if you start changing the stimulus, and this is a relatively low-dimensional stimulus space for sampling, just a 2-dimensional stimulus space, the network's unable to cancel, to learn to balance all of the inputs in this 2-dimensional stimulus space, but it can still semi-balance these inputs. So the network naturally finds this detailed semi-balanced state in which every neuron in the network is in a semi-balanced state. Okay, so let's explore this state a little bit. So this is these are simulations from the same network I showed on the previous slide. And this detailed semi-balanced state, we have plasticity turned on, inhibitory plasticity turned on, except now we started sort of exploring as we trace out, remember this is a 2-dimensional stimulus space, as we trace out this 2-dimensional stimulus space, which now we're tracing it out on a lattice instead of randomly sampling the points. What is the representation of firing rates of these stimulus? And we can explore that by looking at the neural manifold. So as we explore this 2-dimensional stimulus space, how do the firing rates change with respect to one another? And this is just 4 of the astronomical number of neural manifolds there are in a network of 5,000 neurons. And we see that they are nonlinear, and they're highly intricate, right? So you can get all sort of, it's kind of fun actually to play with these simulations, because you can just plot all these neural manifolds and see all these sort of exotic looking shapes. So you get these complex, intricate nonlinear neural manifolds in the semi-balanced state. To quantify how nonlinear there are, one approach is to apply PCA versus isomap. So PCA finds a linear manifold that captures the variability in the data, whereas isomap can find a nonlinear manifold. The amount of variance explained by PCA versus isomap will be different if the representation is nonlinear. And indeed, we see that the PCA needs around 8 dimensions to capture all the variability, whereas ISOMAP captures it in 2, which is what we would expect because there's a 2-dimensional stimulus. So the neural manifolds are necessarily explained by 2 dimensions. It's just a question of whether ISOMAP finds that 2-dimensional representation, and it does. Okay. Finally, we wanted to test sort of the power and the potential utility of these nonlinear representations in the semi-balanced state. So we took the same 2 population network, one E population and one I population, and instead of giving it sort of random stimuli, we gave it stimuli that were projections of MNIST images. So the actual pixel representations of MNIST images, we just projected them onto the excitatory neurons in the spiking network. And what we found then is if we look at the firing rate representation, now we can take these excitatory neurons and look at their firing rates. They're going to have, there's one vector, one NE dimensional vector for each MNIST digit. And let's look at the representation of those MNIST digits in the excitatory neurons firing rate space. What we find is that in this firing rate space, I'll kind of go through this quickly because we're running a little low on time. In the firing rate space, we find that this representation is linearly separable. So we can train a linear readout of the firing rates that perfectly classifies the 10 different MNIST digits. But if we try the same linear classification in pixel space, it doesn't succeed perfectly. We get some discrimination, but there's some misclassifications. That is to say, this nonlinear representation helps with the classification of MNIST digits. And this isn't necessarily surprising. A random nonlinear representation, we have actually found, now that we're addressing reviewer comments, a random nonlinear representation does just as well. But this is just to demonstrate that the nonlinear representation implemented by these networks are actually doing something potentially useful. And to take that one step further, we trained another spiking network, or we implemented another spiking network that reads out the correct labels for these MNIST digits by training this linear classifier and then setting up a winner-take-all network. Don't really have time to go through it, but you can see here that the layer 2 raster plot indicates that the layer 2 is spiking in response to the correct digits, basically. The layer 2 population that spikes the most corresponds to the population with the correct label. Most of the time, it gets some of them wrong. Okay. So just want to make one conclusion. I'd argue that this is sort of somewhat preliminary work in the sense that I think it's a complete work, but there's a lot of directions to go from here. So the main conclusion I want to draw is that not so weakly coupled networks that are large N, and that have time-varying stimuli naturally operate in a semi-balanced state. And firing rates in this state are approximated by this equation. And this is a useful equation in the sense that so the universal word in my talk title is to say that this is model independent. It doesn't matter what type of models you're using for your neurons. This should give you a relatively good approximation and not so weakly coupled networks. And I think there's a lot to explore here. So this is sort of just a first study of these ideas. And yeah, I'm happy to talk more about different directions to take this. My current graduate student, Vicky Zhu, is extending this to networks. Well, I guess these already have plasticity, but what I meant to write here is with learning. So looking at how these expressions can help us understand how to train spiking neural networks to actually learn useful tasks. And if you want information, more information on this, you can see our bioRxiv paper right here. And thank you for listening.
Thank you. Very interesting talk. So we'll have time for one question. And so what we're going to do is the questions also, there were a lot of questions from previous talks as well. They're all on Slack right now. So I hope that Juliana will have some time to answer them on Slack. So there's this is an ICMNS Slack team with different channels. There's a channel devoted to this particular session, and the questions are there right now. So Robert, how were the MNIST digits projected to the E neurons?
So we tried both a random projection and a projection in which, so there's, We took a subset of the MNIST pixels. So the images are 20 by 20. So there's 400 pixels and 4,000 neurons. So we did one example where each pixel projects exactly to 10 neurons. And we did another example where we just multiply them by a random matrix. And we get similar results either way.